Right, please, everyone, so we can start. So today is the last week that we working together on the topic of genetics. In last week, in last chapter, we have working on the Mendelian inheritance in humans. And for the time being, I want you to spend the last week to discuss on epigenetics. So on this part, the human Mendelian mode in disease, I want you to work at home on this topic, okay? So on the pedigree symbol and all the disorder related to the autosomal dominant, autosomal recessive, the X-linked and the dominant and recessive. You can read about it at home. Okay? Just remember that for the family pedigree tree of autosomal dominant, we will have the carrier disorder, the affected patient will be present of X in every generation of the family pedigree. So, for example, in this pedigree tree, we have the affected presented in generations 1, 2, 3, and 4. And with the family, which both parents are not affected, then the family pedigree from this point to the next generation will not showing up the disorder. Okay? For the about the Huntington disease, you can read about it at home. For the autosomal recessive pedigree, we will not observe the affected in 8 generations, but we will see in several families, family trio, will have suddenly both parents are not having disorder, but one of the offspring carrying the disorder, and the ratio of having a disorder are the same between male and female. On the X-linked recessive, we'll see that in each of every family pedigree is not show up in the pedigree, but some in each will show up in only several generations, and most of the affected are male, and the female, which is the parent of this affected individual, and the carrier. That means they are heterozygous and they carry one allele of the disorder. And this disease will show up only once this carrier, the baby, carry the X chromosome having disorder, get the combined with the Y from the father. All right. So now let's talk about genome editing tool. The genome editing tool actually have been researched from 1990s, but recently scientists have indicated very specific mechanisms of how we can interact and interfere in the exact positions of the genomes. Quiet, please. Right? So this discovery having awarded the Nobel Prize in Chemistry in 2021. And this discovery promised open a new huge achievement in not only to do research what is the function of each region of the genome, but also how to generate a new individual without the disruption. Normally, with a carrier, when you're having a genetic disorder, you will have to live with it to the rest of your life. Okay? But with this tool, people hope that we can interfere with the monofunctional fragments of the genome to cure it. So first, let's watch how the genome editing tool was discovered.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus associate, they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognize that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is prepared after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known, but it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider, and this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually there's an important precedent for such a pause from the 1970s when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer, this is a technology with huge consequences you pointed out. Your attitude about asking, you call it a pause or a moratorium or a quarantine, is incredibly responsible. There are, of course, the therapeutic results of this, but then there are the non-therapeutic ones, and they seem to be the ones gaining traction, particularly in the media. This is one of the latest issues of The Economist. Editing humanity is all about genetic enhancement, it's not about therapeutics, right? What kind of reactions did you get back in March from your colleagues in the science world when you asked or you suggested that we should actually pause this for a moment and think about it?
My colleagues were actually, I think, delighted to have the opportunity to discuss this openly. It's interesting that as I talk to people, my scientific colleagues as well as others, there's a wide variety of viewpoints about this. So clearly it's a topic that needs careful consideration and discussion.
So there's a big meeting happening in December that
Correct.
you and your colleagues are calling together with the Academy of Science and others. What is your expectation? What do you hope that will come out of the meeting practically?
Well, I hope that we can air the views of many different individuals and stakeholders who want to think about how to use this technology responsibly. It may not be possible to come up with a consensus point of view, but I think we should at least understand what all the issues are as we go forward.
Now, colleagues of yours like George Church, for example, at Harvard, they say, yeah, ethical issues is basically just a question of safety. Test and test and test again in animals and in labs, and then once we feel it's safe enough to move on to humans. And so that's kind of the other school of thought, is we should actually use this opportunity and really go for it. Is there a possible split happening in the science community about this? I mean, are we going to see some people holding back because they have ethical concerns and some others just going forward because some countries under-regulate or don't regulate at all?
Well, I think with any new technology, especially something like this, there are going to be a variety of viewpoints. And I think that's perfectly understandable. I think that in the end, this technology will be used for human genome engineering. But I think that to do that without careful consideration and discussion of the risks and the potential complications would not be responsible.
Now, there are a lot of technologies in other fields of science that are developing exponentially, pretty much like yours, and thinking about artificial intelligence, about autonomous robots, and so no one seems, aside from autonomous warfare robots, nobody has seemed to have launched a similar discussion in those fields and calling for a moratorium. Do you think that your discussion may serve as a blueprint for other fields?
Well, I think it's hard for scientists to get out of the laboratory. Speaking for myself, it's a little bit uncomfortable to do that. But I do think that being involved in the genesis of this really puts me and my colleagues in a position of responsibility. And I would say that I certainly hope that other technologies will be considered in the same way, just as we would want to consider something that could have implications in other fields besides biology.
Jennifer, thanks for coming to that.
So to use the tool efficiently, we need to know about one information that is critical for the whole process, which is the genetic information. The human genome has been sequenced since 2003. We finally got the full genome sequence in last year, 2022, when we finally tackled the last 8% of human genome, which composed mostly repeatations. However, sequence the human genome is just one part. It is the easy part. The most challenging, time-consuming, money-consuming, and labor-consuming, and much more difficult is annotate the genome, to know which part of the genome to perform which functions. So, for example, based on this technology, CRISPR, for example, you want to edit your offspring. You say that, oh, I want to have children with blue eyes or blonde hair, because you just simply hate black hair. You see black hair in your grandparent, your grandparent, and you suddenly hate the blonde hair. Simply hate the dark hair. You want your offspring must have the blonde hair in nature. So you perform to editing the genome of your offspring. But the detail is, we know which enzyme responsible for the color of the hair. But we don't know that if we modify those genes, those enzymes, whether it's affect only to the color of the hair, but also, or it will affect to other stuff too. For example, the enzyme network system, or the protein interaction in the body, and thus leading to the unexpected result or not. And this question also leading to today's chapter, which is epigenetics. So what is epigenetics? epigenetics actually a science in genetics that research not on the coding of the genomes, but on the how a gene was affected by the environmental factor. In other words, if we consider that DNA is like a blueprint to build a house, actually the genome in a cell not contain only the genetic information to build that cell, but to build another cell. Or we say that it's a blueprint to build a city. And each house must locate where is the information for my house in that pool of information. So during development, we know that for a diploid cell organism like a human, we are developed from one cell, the zygote cell, the first stem cell, which is most powerful cell. We can be differentiated to the different type of cell. But the question is, in human, we have in adults, there's around 1 billion cells in an adult cell. And the cell type is around 265 cell types. And this cell type might have a very different morphology and functions. For example, we have the heart cell, which is different with the brain cell, different with the intestine cell, different with the skin cell, different with the blood cell. So the question is, why it developed from the same cell, the zygote, right? And all the cells in the human body will have the same genetic information. Why we have the morphology of each cell is so different like this? All the cells in the body have the same genetic information, but we have the phenotype, the morphology of each cell type is really, really different. So, for the embryonic development, we have from the first stem cell, the zygote, after around 24 hours, that means after 1 day, it will divide develop into 2 cell stage. After another 24 hours, it will develop into the 4th cell stage. Another day, develop into 8th cell stage. So after 4 days, it will come into the 16th cell stage, and in 5 days, it will develop into the 32nd cell stage. But from the 16th cell stage, the cell will develop into 2 cell types. The inner cell, which is more remarked at the orange color here, and the outer cell, which is denoted as the blue color here. The outer cell later will develop partly into the placenta and partly into the nervous system. And the inner cell will develop into most of the organelles in the body. The inner cell in the development will continue to differentiate it into different 3 main layers. The endodermis, the internal layer, which will develop into the lung cell, the spleen cell, the thyroid cell, and the digestive system. The middle cell, also called the mesoderm, will develop into the bone cell, the muscle cell, the cardiac system, and the blood system. And the outer cell, which is the ectoderm, in the inner cell will develop into the skin cell and the neuron system. So we see that from the first cell, from the zygote, after 4 days of development, we start to have differentiations. So what is differentiations? And how it can transfer one cell to different cell with different morphology. All these genes, all these cells will have the same genetic background. The genetics are the same. The blueprint are the same. But how it can develop into different cell states like this. So when scientists going further to investigate which gene was turned on and which gene was turned off during differentiation, they find out that in a subset of cell, like the cell line, depend on the cell line, a subset of gene was turned off and the other gene was turned off during development. For example, from the zygote, when it develops into the neuron cell, the subset of genes related to express dopamine is turned on during development. In the other hand, the subset of genes related to express the hemoglobin and the myoglobin was turned on during development. On the other hand, with a cell that produce the blood cell, all the genes that related to express the hemoglobin will be turned on, and the subset gene related to dopamine and myoglobin will turn on during development. Quiet please, everyone, quiet. In the muscle cell, in the other hand, the subset of gene related to hemoglobin and dopamine was turned off, and the subset gene related to express the myoglobin was turned off during the development. And this is referred to a term epigenetic marker. What is epigenetic marker? It's the marker that denote that which gene was turned on and which gene was turned off during the development of the cell. Now, talking about epigenetic marker, first let's talking about stem cell. What is stem cell? So stem cell is the type cell that can differentiate to a specific type of cell line. We have 5 most popular types of stem cell. The most powerful is a pluripotent stem cell. This cell line, like the zygote, can basically differentiate it to any type of cell in the human embryo. And of course, with this unlimited ability, it will not have the epigenetic marker restrict less the most. The second type of stem cell is pluripotent stem cell, which is developed in the after 6 or 7 days of the embryonic development. This can be divided into different game layers: ectoderm, the endoderm, and the mesoderm. Basically, it can still differentiate to a wide range of cells in the embryonic development. The next type is multipotent stem cell. Multipotent stem cell can differentiate to a certain type range of cell line. It's not powerful as pluripotent and multipotent stem cell, it can only be differentiated to a certain site. And the next one is the oligopotent stem cell, which it can feel differentiated to several cell lines that's closely related. And the last one, which is also the least powerful stem cell, is the unipotent stem cell. Uni, that means that it has the capability to differentiate it to only one cell. Okay? So during development, we see that study about epigenetic in differentiations sometimes are really similar, like when we study about twins. Okay? Anybody here have seen the twins? I mean, monozygotic twins. Anyone? Anyone have friend or observed a monozygotic twin? Yeah. So, if we observe a monozygotic twin, we can realize the fact that when the twin was small, they are really hard to distinguish between each other, right? Even their parent cannot distinguish which one is which one. But when they grow up, especially when they are over the age of 30 or 40s, we can easily to separate or distinguish which one is which, right? You see this fact? So, during development, when we study about monozygotic twin, we see that they are really look alike when they were born. But come along when they are grow up with a different lifestyle, with a different environmental factor, when they grew up, their genomes are going to be different. So for example, in here, scientists have studied the difference in the epigenetic marker in the chromosome number 3 of the 3 years old twin and the 30 years old twin. And they mark that one individual with the site 3 and the other with the site 5. Site 3 is have the green color and site 5 is have the red color. When they match the chromosomes of the two twins, if the epigenetic mark the same in between the two twins, we'll see the yellow color, which is the combination between the red signal of the site 5 and the green signal of the site 3. Once they observe that one signal are stronger compared to each other, for example in here, the red color, that means that the epigenetic mark in this one, in these regions, are stronger than the other one which is marked with the site 3. And the other green part here, that means the site 3 is stronger than the site 5. So when they compare in the chromosome number 3 of the 3 years old twin, they see that the epigenetic marker of the two twins are more or less the same. We hardly find any differences in the expression level between the 3 years old twin. But in a 50 years old twin, they see that more and more part of the chromosome 3 are more or less expressed different between the two. This publication publication was published in 2005. They see that although the twins are epigenetically indistinguishable during early years of life, that means when they are small, when they're just born, the older monozygote twins exhibit remarkable differences in their overall content and genomic distribution of the 5-methylcytosine DNA and histone acetylation, which is affected into their gene expression or trait. That means the older they grow up, the more difference in the environmental factor that can cause into the epigenetic marker, which can lead into the differentiated of the two twins. Study about the twins, scientists continue to find out that even the monozygotic twins have the same genetic background. 30% of the twins will have different outcome in height. And sometimes the differences is really critical, like in this case. So, in this case, yes, if you are wondering, this is an identical twin, the monozygotic twin. But we can easily find out, clearly distinguish which one is which, right? Because they are different in the height, in the percentage of muscle, and so on. So, genetic factor is just one thing to control. So, what is the mechanisms behind this? To answer this, scientists in Utah University have built an experiment in the mice. So in the mice, they have built two cell-like mice. There are two mice at the same line age. And in the turn, at the same line age, that means these two mice have the same genetic background. Okay? They have the same genomes, not different with each other, even in a single nucleotide. Okay? These two mice are grow up in the same conditions of temperature, same condition of humidity, same conditions of atmosphere pressure, same conditions of physical activity. Almost all the conditions of development are the same, only one difference. In one mice, they're using a comb, like a comb you usually to brush your hair. And they're using the comb to brush the fur of the mice. In the other mice, they didn't do it. Okay? So the two mice, same genetic background, same genomes, same conditions of development, only one thing differences. In one mice, they're using the comb to brush their fur, and the other, they do not do it. Okay? You understand the condition of the experiment? So, they start to observe how these two mice reacted when they grow up, with only differences like this. And they see that the highly nurtured, that means the mouse that we use in the comb to brush the fur, when they grow up, they become really relaxing and calm mouse. The heartbeat is stable, and when they heard an unexpected noise, they heard to alert to the noise to see that if there is the noise is a signal of dangerous, and if it is, they ran away. The other hand, the mouse that the scientists do not use the comb to bust the fur. When they grow up, they become so anxious, always fretful, always nervous. Their heartbeat is so fast. Always like this. And when they heard an unexpected noise, they immediately run away. They don't care if it's a signal of dangerous or not. They immediately run away. They seem always nervous, and they have a significant shorter lifespan compared to the common mouse. When the scientists researched about this case to see what mechanisms behind the different behavior in those mice, they find out a gene called GR gene. GR is stand for glucocorticoid receptor. So this gene is expressed strongly in the brain cell, and it provides the GR protein to calm down the mind. It works like morphine. It calms down the mind. With the high neutral, that means when the scientists, the mouse, calm, using the comb to bust the fur, like the behavior of the mother mouse, they always licking their fur, the fur of their offspring. Yes. And the more the mother mice licking the fur of the offspring mice, this GR gene will start to stretching out. And when it's stretching out, it creating space. So the transcription factor will have a space to binding in the gene is starting to synthesize protein. The other hand, the anxious and stressful mouse, the GR genes was not stretching out, and it created not enough space, so the transcription factor are not coming by and start to synthesize the GR protein, making the mice always stressful, always nervous. The most interesting thing is, this process only happens when the mice are under adult age. That means when the mice are developed into adults, no matter how much the scientists calm the fur, the GR gene are not expressed out. Okay. And this story is a little bit similar with the story of the dolly ships, right? You all know here, know about the dolly ship, right? It was created in, I remember, in 2000s. So, the dolly ship is created by so the nucleus of the dolly ships were created of the mice, and it will be created put inside a cell of another cell. And this one will be put into worm of another cell to carry to be part of the dolly. But the funny thing is, we have a lifespan of an average 6 is around 12 years. So this is the average shift lifespan, about 12 years old. And the nucleus of the mother cell, when they create it into the dolly cells, so the mother cells is around 6 years old. and scientists creating the nuclear of the cell. And when they're putting into the set and creating the Dalai set, is the lifespan of the Dalai set is only another 6 years, which is significantly shorter than the year of an average six. But if we combine between the lifespan of the mother when scientists create the nuclear and the actual lifespan of the Dalai ship, we will have it's equal to the lifespan of the normal ship. Okay? So what is the story behind this? The story behind this is even we trying to create a zygotic, a new monozygotic to develop into the embryo, something beyond the genome system will still there to remark it that this is the cell of the 6 years old sip. It's not the zygote sip when the year calculation is just 0. Okay? Now that we know that it's called the cell memory, or sometimes the cell memory. Or nowadays we know that is the epigenetic marker. Okay, so now we will have a small break, a 5-minute break before we go further on the mechanism of epigenetic. Okay, 5-minute break. Okay, quiet please. Everyone, quiet please so we can start. Quiet please. So we are talking about ownerships. We are talking about difference in the development of the monozygotic twins. We are talking about a different phenotype and morphology of all cell types in human body which have the same genetic background because it developed from the same cell. All this evidence showing that there is something beside the genetic factor to control which gene was turned on and which gene was turned off during development. Okay? So genetics, epigenetics, is the study of the change in the regulation of gene activity. And this regulation is not dependent on the sequence of DNA, but it's dependent on the environmental factor or lifestyle. So there's something design of the sequence of DNA, A, G, G, and T, and control what is the differences in the expression level of each gene. So epigenetics is the science to study about the expression level of genes. And if we have in the DNA, which is referred to transcription and translation, to the consequences of how the gene will express the phenotype, we have the term genome to refer on the whole gene set in the individual. Then in epigenetic, we have also the term epigenomic is to refer to the global scale in the whole genome, which gene was turned off and which gene was turned on. Now there's many different mechanisms of epigenetics, but in our training sessions, due to the limitation of time, I can only introduce you three main basic mechanisms of epigenetics, which is DNA methylations, histone modifications, and RNA, which is the interference RNA. Okay. So first, let's talking about DNA methylations. So what is DNA methylation? So DNA methylations mostly refer to the methylated of cytogens in the genome. The cytogens, this is the structure of the base cytogens. You have 4 type nucleotide, right? 4 type corresponding to 4 type of base lipid: adenine, cytosine, guanine, and thymine. And the DNA methylation was mostly happened in cytogens. The cytogen in this case will have another methyl group adding in the carbon here, carbon number 4. And normally, in the genes, in the 5' upstream region of genes, and most of the cytogens was methylated. That is a signal that to show that the gene was untranslated. The gene was turned off. For example, in here, we have a CG here, which is methylated. So, in the beginning region of the gene, we have the exons, first exon here, and this is the promoter region. Mostly the 5' upstream, including the promoter regions, and part of the exons was methylated, so the gene was turned off. And if the genes, the beginning of the genes was methylated, that means the gene was turned off during development. Now, DNA methylation was happened mostly in the region called the CPG island. CPG island is a term referred to a sequence that rich with C and G sequences. For example, we are seeing here a sequence in the beginning of the genes. We can see A, T, G, the starting codon here. This is the translation starting site, translation starting site. So we see that here is the first exon, and you can see that the ratio of C and G will appear very frequently. The CPG is very frequent in the beginning of genes, and if the CPG island was mostly methylated, that means it's a signal to see that in this gene, this song, the gene was turned off, was untranslated during development. Now, the mechanisms DNA methylated would happen. And this is also leading to the how the cell memory can be passed through from generation to generation. So when the cell will under mitosis, that means it will go into the cell division and produce two or other daughter cells with the genetic background are the same. It also keeps its cell memory, or the methylated level of the cell was keeping the same. Okay? For example, in here we have double-strand DNA undergo mitosis, and we have the two C, G, the C inside the kit was methylated here. When it's divided, it will continue with an enzyme called DNMT1, and this DNMT1 will methylate the C here and create that the DNA methylated will be maintained during mitosis. Now the DNA methylations was play many roles in mammalian cells. For example, X-inventing or X chromosome inactivations, and control the development of the cell, maintenance the heterochromatin, that is say the chromosome structure development. Now let's talking about one example of DNA methylation, which is the X chromosome inactivation. So you all know that in most of the mammalian cells, the X is the gender chromosome X is representative the female, and the XY is represent the male. Now the X inactivation is caused mostly by the DNA methylation of the CPG island during the X. So you all know about the calico cat, right? We know that the calico cat is the female because the gene that corresponding to the color of the fur is located in the X chromosome. But the question is, why we have all the cells in the calico cat have the same genetic background? That they are both heterozygous of the gene corresponding to the color of the fur. Why we have in the calico cat part of the fur with yellow and part of the fur with black, although they have the same genome. Chưa hiểu câu hỏi. Okay. Các bạn đều nhìn thấy mèo tam thể rồi đúng không? Học ở cấp 3 rồi, mèo tam thể chắc chắn là mèo cái, đúng không? Bởi vì là gen quy định màu lông là nằm trên những cái thể X. Bây giờ câu hỏi của tôi là tại sao trên con mèo đấy toàn bộ cái tế bào trên cơ thể con mèo đều có kiểu gen như nhau, XA lớn XA nhỏ, và tại sao có một đám lông ở chỗ này thì nó lại vàng, một đám lông chỗ khác thì nó lại màu đen, mặc dù tất cả các tế bào lông đấy đều có một thể chung một thể genome, đều là XA lớn XA nhỏ cả. Hiểu câu hỏi không? Đúng không ạ? Tất cả kể cho dù là mèo hay con người thì đều đầu tiên là đều phát triển từ một tế bào trứng từ mẹ, một tế bào tinh trùng từ bố kết hợp với nhau tạo một hợp tử, và tất cả tế bào trong cơ thể đều phát triển từ hợp tử này. Như vậy tất cả tế bào trong cơ thể đều có chung một hệ gen, hay nói khác là cái background, cái DNA của tất cả các tế bào trong cơ thể là như nhau. Tại sao? Tế bào tim nó khác, tế bào da nó khác, tế bào xương nó lại có hình thái khác, tế bào máu khác. Hay trong trường hợp của tế bào mèo tam thể thì tại sao một nhóm tế bào lông thì nó lại biểu hiện màu vàng, một nhóm tế bào lông thì nó lại biểu hiện màu đen? đều là kiểu gen điện tử cả, đúng không? Kiểu gen tất cả tế bào đều là điện tử tác động. Hầu như rất hiếm. Okay, so this is explanations. It's because of the X index activation due to the DNA methylation. So one thing about X inactivation is happen randomly. Okay. During development from the first zygote, it will continue to divide through mitosis to develop into a group of cells. Like in human, we have the 2 cell stage, the 4 cell stage, 8 cell stage, 16 cell stage, 32 cell stage. But in come along with the development of the number of the cell, it starts to differentiate differently, and leading to consequences that in different cells, one X will inactivate it. And which X was inactivated is choosing randomly. For example, let's say in the paternal, this X is corresponding to the black fur, and the maternal, this X is corresponding to the yellow fur, right? During development, the cell with mitosis develop into different cell lines. X will have the heterozygous. One X will correspond to black from the father, and one X corresponding to the yellow from the mother. But in one group of cells, the X from the father was keeping unharmed. Meanwhile, the other X from the mother will be inactivated by the DNA methylation. So leading to consequences that all the genes on the X chromosome was silent. And if this gene X was corresponding to the yellow, then in this group of cells, it will show up the black color because the yellow was silent. In the other group, now the silent X is come from the father. So in here, the expression genes was come from the mother, which had the yellow first. And this group with the X from the father was silent will develop into the yellow color. How about the white? Well, the white, it comes from another phenomenon that we have studied in chapter 5, which is epitosis. Epitosis is another, this PrEP gene was controlled by another PrEP gene was located in the same chromosome X, and if it expressed out, it will eliminate the activation of the both 2X and create the white color of the cell. The DNA methylation are also show up when we compare between the antigen marker between the pluripotent cell, that the stem cell that can differentiate into different types of cells that we discussed in earlier discussions, and the unipotent cell. You can see that this is a sequence genes in the beginning of gene. The two sequences between the pluripotent cell and the unipotent cell, the DNA sequences are the same. All start from CTG, all the same. This is a beginning of gene. You can see the ATG here. So this is a translation start. And the first, the transcript starting site happens right here. So this is the reason of the first exon. And all the reason is 5' upstream increase. We can realize the CPGI was hidden in the beginning of the genes. So basically, the DNA sequence are the same. But when we compare the DNA methylated between this pluripotent cell and the unipotent cell, we'll see that most of the CPG island of the unipotent cell are methylated. Meanwhile, in the pluripotent cell, the CPG island was unmethylated. So that signal shows that industry was turned off in the unipotent cell, and it will be turned on in the pluripotent cell. So in differentiation, the different factor in the environment will decide to turn off these genes when the pluripotent cell differentiated into the unipotent cell. Now let's see small demonstration on the DNA methylations in the cancer cell.
In the cell, there are many methods for controlling how DNA is read and expressed. Several of these methods, called epigenetic modifications, can turn specific genes on or off without altering the DNA sequence. One of these methods is DNA methylation, which occurs when a methyl group is added to a DNA base. When this happens, additional proteins can no longer bind to this region of DNA. This prevents the DNA from being transcribed to RNA. The gene is turned off. Normal methylation patterns are important for the development and function of healthy cells. In cancer cells, abnormal DNA methylation patterns are seen. Genes that would normally suppress tumor growth see higher levels of methylation and are turned off. At the same time, genes that increase tumor growth, called oncogenes, see a decrease in methylation and are turned on. This results in unregulated tumor growth.
I come to the second mechanism, which is the histomodifications. So we all know that in eukaryotic cell, DNA was associated with the complex protein called histone, right? And the complex DNA and histone protein form the structure of the chromosomes. So DNA is one three-quarter into the nucleosome called the octamer. So this is the octamer. The octamer is composed by 8 subunits, composed 2A2A, 2 subunits of A2B, 2 subunits of A3, and 2 subunits of A4. However, histomodification related to the tail of the octamer. We have a tail here. And if, for example, in one subunit group, we have the acetylations. The acetyl group is the group that pushing positive electronic in the electron core. So if you put the electron in the nuclear shell. As a result, because the acetyl group will put the electron in the intermediate core, we will have the nucleation core now will have a negative charge, right? And DNA have negative charge under the neutral pH because DNA was composed by nucleotide. Nucleotide composed by three main groups: the 5-carbon sugar, the base nitric, and the phosphate group. The phosphate group will have a negative charge, right? So the DNA strands have a negative charge. The nuclear core also have a negative charge. As consequences, the DNA will lose dance against the nuclear core and create more space, so that the transcription factor can bind in and start to transcribe the genes. So normally the acetyl group of the histone core is marked for the turn on, the activated of the genes. In the other hand, if we have the methyl group with aspect into the histone core. Now the methyl group is the taking electron out of the nuclear core, which is make the nuclear core have more positive charge. Okay? The more positive charge, DNA is negative charge, and the nucleus core is positive charge, will make the DNA is more dense against the nucleus core, which is make the transcription factor less space, becoming and binding into the gene to start the transcription process. So this whole process is composed by different modifications, the acetylations, methylations, ubiquitinations, and summilation and phosphorylations. And this process was carried by a different enzyme system. So for example, if we have a modification happen here in the lysines of 27 in the histone tail protein, we'll have the mark, which is this is the symbol that published in 2011 in histone number 3 in the amino acid lysine at the position 27 is acetylated. And we'll see the difference of histone modification was take place by different enzymes. For example, the acetylation was carried by H18, which stands for histone acetylation transferase. And the deacetylation was carried by the H dot, which stands for histone deacetylated. The methylation and demethylation also was corresponding by different enzymes, HMT, histone methylated transferase, and histone demethylated enzyme. Okay. Normally, if we have the acetylated, for example, in here, we have the blue ball is octamer of the histone core, and the acetylated was unexpressed because of the enzyme histone deacetylated was removed the acetyl group in the tail of the histone, which is linked to the positive charge, the more positive charge of the nuclear core, which is linked to the transferring factor can have enough space to start to transfer the beads. Meanwhile, if the process will take care about the HAT, histone acetylated transferase, it will provide the more negative charge in the histone, leading to the more space to the transferring factor binding and start transferring. Let's see a demonstration about histone modification.
Epigenetics refers to the heritable modifications in gene functional activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around the core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
Okay, come to the last mechanism of today is the interferon RNA. So DNA methylations and histone modification combine together to create what is so-called the epigenetic marker or semen. But interference RNA is interact to control transcript at a different level. So with the normal gene, when it happens, when the transcription happens, the fragment of DNA will be open and the transfer factor and start to transcribe to create the mRNA. The mRNA been through the mature process to adding 5' cap and poly A tail and remove the intron and getting out of the nucleus region through the hole in the nucleus membrane into the cytoplasm. And the translation was happen in the cytoplasm when the ribosome start binding in the mature mRNA and start to synthesize protein. We have discussed this in chapter 3, right? However, in the iRNA, it will interact at the level after the control mRNA can be sent out of the nucleus region in the cytoplasm. In detail, the iRNA was transcribed from a fragment of DNA, that the fragment that we don't know the function yet, which is not coding for protein. But this combined with a complex called RISC and DICE, which will remove or cut and erase the mRNA that have the sequence matched with the sRNA here. And while the machinery with the guided mRNA will catch with the mRNA have the sequence of the DNA, it will immediately cut off and then eliminate the delivered mRNA out of the system. And no mRNA, no translation, no protein product from the system. Okay?
RNA interference was popularized by work in C. elegans, when long double-stranded RNAs were injected into a worm's gonad, a standard way of introducing transgenes into worms. They blocked the expression of endogenous genes in a sequence-specific manner. In eukaryotes, most protein-coding genes are transcribed by RNA polymerase II, which generates pre-mRNAs that are then processed to form mature mRNAs. These mRNAs are then transported from the nucleus to the cytoplasm, where they are translated. RNAi can regulate endogenous gene expression. RNAi can be set in motion by genomically encoded short regulatory RNAs known as microRNAs. In algae, worms, and flies, RNAi can be activated by endogenous transposition. In plants and cultured insect cells, RNAi also has a role in antiviral defense, in which viral double-stranded RNAs are targeted for destruction by the RNAi machinery. When long double-stranded RNAs enter a cell, they are recognized and cleaved by dicer, which is a member of the RNase 3 family of double-stranded RNA-specific endonucleases. Cleavage by dicer creates short double-stranded RNAs that are characterized by 2 nucleotide-long 3' overhangs. These are called small interfering, or siRNAs. siRNAs can form a ribonucleoprotein complex called RISC, or RNAi silencing complex. This complex includes slicer, an argonaute protein with an RNase H-like domain called PWE. RISC first mediates the unwinding of the siRNA duplex. A single-stranded siRNA that is coupled to RISC then binds to a target mRNA in a sequence-specific manner. The binding mediates target mRNA cleavage by slicer. The site of the cleavage falls in the middle of the region of siRNA complementarity. The cleaved mRNA can be recognized by the cell as being aberrant and then destroyed. This prevents translation from occurring, silencing the expression of the gene from which the mRNA was transcribed. In plants, the aberrant RNA that results from the RISC-mediated cleavage can also serve as a template for RNA-dependent RNA polymerase, or RDRP. This process relies on unprimed RNA synthesis, in which the aberrant RNA is used as a template. The resulting double-stranded RNA is a substrate for dicer activity, which generates more siRNAs. In some organisms with endogenous RNAi mechanisms, for example, fungi, plants, worms, and mammals, RNAi also involves another amplification step. In this step, single-stranded siRNAs not associated with RISC bind to their target mRNAs in a sequence-specific way and serve as a primer for RDRP to polymerize the antisense RNA strand. Such specificity is intrinsically sensitive to natural sequence variation. The double-stranded RNA molecule that is created serves as a substrate for dicer, which cuts it into siRNAs. In turn, these can either unwind and prime RNA-dependent RNA polymerization, or together with RISC, mediate the cleavage of target mRNAs. This amplification, coupled with RNAi spreading between cells, is thought to underline germline transmission of RNAi in worms. RNAi spreading has also been described in plants, but not in mammals.
Okay, so the demonstration about RNA has finished with chapter 7 and also closed all the session on genetic cost. Do you have any questions? Any questions? Okay, then in each generation study genetics, I always ask you, do you have any comment about the cost? Whether the cost is taught too fast or too slow, and the knowledge involved in this course is too difficult or too easy for you, please let me know. Okay? Whether in the class you cannot absorb the knowledge because of the content of the lecture, or because of my English level, or because of the facility system, please let me know. Any comment? Any suggestion I can make for the new generations?
Em xin phép tiếng Việt nhé. Thực ra thì về cái course genetics này thì bản thân em thấy em cũng là một người khá là yếu thần sinh. Tuy nhiên là với thầy thì thực sự em thấy thầy làm quả thật là một giáo viên rất là tuyệt vời đối với em luôn. Thầy cái cách nói thật kiểu thầy lấy lại gốc sinh cho em luôn ạ. Và cái nội dung giảng dạy của thầy đủ ngắn gọn, đủ ấn tượng và nó đủ khả năng để influence em. Nên là các tiết học của thầy thì em không hiểu thế nào. Và rất là cảm ơn thầy đã cho em một tiết học ý nghĩa ạ.
Buổi cuối cùng này thì em với lại tất cả các bạn thì xin gửi lời cảm ơn tới thầy đã giảng dạy cho em nhiều kiến thức bổ ích, và bọn em sẽ luôn ghi nhớ công ơn thầy. Em cảm ơn.
Any other tips for the learning material that I should make an improvement for you?
Về cái slide này thì ở trong lúc đầu thì nó hơi thiếu ạ, thì em nghĩ là cái thì thầy có thể xem xin lại, và cái lần sau thì thầy sẽ apply tốt cẩn thận hơn. Thì em nói thêm một điểm nữa, đó là pass của em thì là bọn em phải qua cái cellular biology rồi thì mới học đến cái genetics này. Thì cái cellular biology em cảm giác kiểu nó chỉ đơn thuần là nó là những cái bên ngoài đấy, nhưng mà còn cái bên trong sâu của cái con cell ấy, thì nó liên quan đến genetics, và thực sự là thầy cũng đã làm cho em hiểu sâu hơn rất là nhiều về sinh học ạ. Em cảm ơn.
Thực ra thì môn tế bào thì sẽ giới thiệu về các thành phần của tế bào, trong đấy là giới thiệu về những thể như là cấu trúc của tế bào, bảo quan của tế bào. Tuy nhiên về genetics thì nó sẽ đi sâu hơn về chức năng của genetics. Những cái nhiễm sắc thể đấy nó đảm bảo vai trò để làm như thế nào để có thể truyền thông tin đến thế hệ sau. Thì về cái slide thì mình xin giải thích như thế này là trong slide ấy, khi mà mình trình chiếu trên lớp thì mình có involve thêm một số những cái thành tựu nghiên cứu trong về mặt di truyền những năm gần đây. Thì thực ra những cái kiến thức này là để nói thêm cho các bạn muốn tò mò về di truyền. Những phần này thường thì chiếm khoảng độ 2 đến 3 câu trong 60, các bạn thi cuối kỳ là sẽ có 50 câu trắc nghiệm, thì những phần này chiếm từ 2 đến 3 câu, chủ yếu là để như là một phần thưởng cho những bạn nào mà muốn quan tâm về những cái thành tựu nghiên cứu gần đây. Thế còn cái slide ở trên Moodle nó chứa toàn bộ những cái kiến thức cơ bản mà các bạn cần ôn ở trong cái nếu các bạn chỉ cần thi cuối kỳ để mà có một cái điểm số chấp nhận được, vì là cái hàm lượng cái kiến thức, những cái phần update này, cái thành tựu khoa học gần đây là nó không nhiều, và mình cũng không muốn nếu mình áp lên thì khi OT các bạn sẽ không biết là OT ở đâu cả, giống như là rất nhiều slide và không hiểu ông này ông nói những cái linh tinh gì, toàn những cái vân vân đúng không, biết ôn ở đâu cả. Thì nếu bạn nào mà chỉ cần học môn này qua đâu chỉ cần slide trên Moodle là được rồi. Còn những cái phần mình giới thiệu, ví dụ như là cái clip đầu tiên nói về cái CRISPR-Cas9, thì thực ra CRISPR-Cas9 thì mới được biết đến rộng rãi từ năm 2021, và nó được giải Nobel vào năm 2021 và được sử dụng rộng rãi vào những năm đầu của thế kỷ 21. Nhưng nó cũng không gần như chắc chắn là phần CRISPR sẽ không có trong phần thi cuối kỳ, vì nó một cái thành tựu của những năm gần đây, cho nên là mình không muốn post tất cả những cái gì mà nóเป็นthành tựu lên trên Google vì nó quá rộng cho các bạn. Các bạn nào có ý kiến góp ý gì thêm các bạn?
Xin thầy gặp link của cái video thì chiếu trên slide còn slide.
Về các link video thì mình đã chia sẻ ở buổi học gần đây rồi, là những cái video clip này thực ra vì lý do bản quyền cho nên mình không thể gửi link cho các bạn được. Tuy nhiên các bạn có thể lên bất cứ một cái web, ví dụ lên YouTube các bạn gõ IR đây, gõ những cái title của những cái video đấy lên là các bạn có thể tìm thấy những cái clip đấy rất là dễ dàng. Còn nếu mà mình gửi link cho các bạn thì phía sau nếu nó leak ra ngoài thì lại là mình đang vi phạm bản quyền của những cái clip đấy, thì nó không hay. Cái này thì hết sức thông cảm. Cái practical chapter là những cái kỹ thuật sinh học phân tử được làm ở trong lab tại UFTH. Thì những bạn nào mà quan tâm hơn thì đến năm 2, năm 3 thì các bạn sẽ được các thầy cô giới thiệu về cái này. Cái practical chapter thì những năm trước mình có giới thiệu cho sinh viên, nhưng mà sau đấy thì mình thấy là một số các bạn mà học ICT hoặc là một số các bạn mà học các chuyên ngành khác thì cái chapter này nó không cần thiết các bạn, cho nên là cái chapter đấy bỏ đi nhé. Nếu bạn nào thực sự quan tâm đến về sinh học phân tử thì đến năm sau thì bạn sẽ được học kỹ hơn về cái practical chapter này. Okay, so since you have no more common questions, I want to thank you very much that you have really patient to attend to my course. I hope that my course is useful somewhere to you. And thank you, and see you at the final test.